Good morning. We are on chapter 18. This is chapter of the blood. This chapter is part of the circulatory group, which is blood, heart, and blood vessels. So we'll start with the blood or the water, they say, and then the blood vessel is the pipes, and the heart is like the pump. All right. So blood itself has been a mystery in the early medical ages. Many myths about blood said it was coming from the air that we breathe, said that it was um, part of us that gets tainted or bad. So in the early times, if you were sick and they look at you and say that you need to get well, they would take blood out of you. Maybe they look at you, how many days you've been sick? Like 10 days? Oh, okay. Maybe we need to take about 4 cups of blood from you, or maybe 5 cups, because that's bad blood in you, and you'll get better. Which, for modern reasons, we know is actually the opposite, because if you take out the blood, you take out the white blood cells, which is what you need in that sickness situation, in that disease situation. So a lot of myths, a lot of postulates, and there were good ones and there were not so good ones. Fortunately, we ended up learning more about the blood, learning more about its origin, its capabilities, and also being able to uh, make it better use for the medical field. As I have mentioned, blood is the first topic that we will discuss. Blood is uh, will also encompass the function of collecting waste in the body, the function of collecting hormones from one organ to the other, delivering it from one organ to the other, the function of providing police or security to areas of your body. Where blood is, white blood cells will make it. Where blood is, platelets will make it in case there's a bleed. At the same time, we are able to take out any acidity, any alkalinity in that organ, and then balance it. Anything that's in excess of acid or base will be taken out through the urinary, through the digestive system, through the respiratory. But blood delivers all these. So you can see how important blood is. It's like having your mailman, your garbage person, your grocery man or grocery person, your probably your gossip person, if you would, all in one package, because it goes around all throughout the body. At the same time, blood also goes through the body in order to cool down some organs. Some organs may be in a level of inflammation. As it passes through that organ and goes out of that organ, it carries with it a temperature. And when the blood uh, goes to vessels on the superficial part of our body, it is able to radiate the heat off and therefore cool the body down. So blood and its the circulatory system as a whole, with blood going through our systems, is a very vital part of our existence. Very vital part, and we also provide to make it be at its best condition, right? Iron, oxygen, nutrition, to be able to keep it at its best. The study of blood is hematology. The specialist, and specialist we add logist, so the specialist in that field is hematologist. Most of the hematologists in practice usually sub train to become oncologists as well, so they can be hematologist oncologists. Our goal for here is for this topic is to know the function of the circulatory system pertaining to the blood, the physical properties and components of it, uh, viscosity of blood and osmolarity and factors that affect it, and some general terms. Circulatory system consists of the heart, the blood vessels, and the blood. If it's cardiovascular system, it's only the heart and the blood vessels. So like we mentioned in the beginning, the functions of this circulatory system is transport, protection, regulation, pretty much what I mentioned at the beginning. And it does that 24/7, 24/7. Even if you're sleeping, it still uh, processes it together with the heart, together with the blood vessels. Adults have 4 to 6 liters of blood, and it will continue to be replenished as the red blood cells reach their maximum uh, lifespan. Blood is a connective tissue, and it also has matrix with it. For this, plasma is the matrix, and the cells, the red blood cells, the white blood cells, the formed elements, we call it, swim, if you would, swim in the matrix. Okay? This is a good picture of a smear. A smear is when you get a sample of blood, like for example, a capillary puncture, and then you put it on a slide, and then you slide it with another uh, glass slide. We do this in the ground class. I let them slide it and be able to look at it in a microscope. But that's okay because now we have a good example in front of us. These are the red blood cells. Where's my pointer? These are the red blood cells as you see it, right? Almost transparent in the middle. And the small stone-like features, okay, are your platelets. Small stone-like features are your platelets, as you can see here. It's highlighted there. While the neutrophils are the one highlighted here. Here, these are neutrophils, and they're highlighted here as well. Neutrophils are white blood cells, right? And all these, okay, highlighted in blue-green, are the red blood cells. So in peripheral smear, you can also see a red blood cell occupied by a parasite in cases of malaria. Okay? That's a very classic diagnostic tool in malaria, peripheral smear. You can also see pictures of sickle cell anemia in peripheral smear, and you can see the actual red blood cell shaped like a sickle. Right? So if there was a parasite, you could also catch it, other parasites, I mean, you could also catch it in a smear. As far as the blood is concerned, we said the matrix was the plasma, and the formed elements are the constituents or cells that are inside that blood. So for the formed elements, we always say red blood cell. It's not only red blood cell. There's many other formed elements, which red blood cell is part of it. So for this, one of the formed elements is erythrocytes, a red blood cell, platelets or thrombocytes, thrombocytes, leukocytes or white blood cells, and for that matter, you have 5 leukocytes in general. 3 are granulated and 2 are agranulated. When you put an A in front of it, that means without granules. So granulated, there's basophils, eosinophils, and neutrophils. Basophils, eosinophils, and neutrophils. Agranulocytes are monocytes and lymphocytes. This picture shows you the platelets, the monocytes, and the neutrophils. You also see the red blood cells and the lymphocytes. Lymphocytes are T and B lymphocytes. So this is basically the formed elements present in your blood. When you take a sample of the blood, let's say from your median cubital vein in the bend of your elbow. This sample can be put in a machine, right, which will spin it. And when it's spun, you will see a separation of the plasma and the formed elements. This process we call hematocrit, or capital H, small letter case C, small letter case T, HCT. Hematocrit will separate the plasma from the formed elements. So basically, what happened was the red blood cells, being the heaviest, will settle at the very bottom. The white blood cell and platelets settle at the middle, forming the buffy coat, and the remainder would be plasma. But in plasma, that includes water, protein, nutrients, electrolytes, nitrogenous waste, any hormone that the blood is transferring, and gases as well. Okay, so this is your centrifuge. That's the vessel. There's your median cubital vein. Okay, and this is part of this is phlebotomy. Otomy means to incise, and phlebo means a vessel. So phlebotomy. So here you have your finished product, which is the plasma, the buffy coat, and the red blood cells. Another tube we call tiger top tube will have a gel at the bottom, but when you spin it, if it's filled with blood and you spin it, that gel in the bottom, which is lighter than red blood cells, will end up at, on top of the buffy coat, therefore sealing the plasma content from the formed elements. Once that is done, it is able to be transported without them mixing again. Usually, lab procedures and laboratories request that you do this if your sample is to be transported, and you can do this in your clinics or in your stations, maybe if you're working with hematology. All right, so let's talk about plasma. Plasma is a liquid portion of the blood. It is called serum if the blood clots and solids are removed, meaning there's no more fibrinogen. It's been used. Okay, so it's identical to plasma except for the absence of fibrinogen. In the plasma, you also have proteins. Number one protein is albumin, produced by the liver, which is used and in transport and also for being able to attract water to stay in the blood vessel. Water, if there's a lot of proteins and there's less water, it will attract water to come in by osmosis, come in and stay in the blood vessel. This is important in our circulatory system because when you are at the capillary level, water goes out at the arterial end and you have to attract it back at the venous end. 85% go back attracted to the osmotic force, but 15% may be left behind because they're too far from the actual vessel. However, but that's okay because the 15% will be collected by the lymphatic system anyway. The lymphatic system ultimately joins the venous system at the end. Uh, Proteins also exist in the form of globulins or antibodies, which is very important for our immune system. And we know that, especially in this present COVID times, antibodies are a hallmark of having gone through the disease and also as a resistance to the disease. There's alpha, beta, and gamma globulins. Fibrinogen is the beginning item needed to produce fibrin in case there's a clot. It's also in the plasma. Plasma proteins are formed by the liver except for the globulins. The globulins are formed by the plasma cells, okay, not by the liver. There will be nitrogenous compounds, amino acids from our breakdown of protein. There will be toxic end products also from the forming the nitrogenous waste, which will end up being removed by our kidneys. There'll be nutrients from our digestive system, bringing it to the liver, and then also bringing it from the liver to the systems. Oxygen and carbon dioxide are present depending on if you are on the venous side or the arterial side. Sodium and electrolytes are also present, especially if you are um, in a near imbalance. The thickness of the blood is viscosity. It is also a factor for resistance to flow. If it's too viscous, it will have a hard time to flow. If it's too thin, it will have a easier time to flow, but the heart will have to compensate by pumping extra hard. Whole blood is about 4 to 5 times more thicker than water, and plasma is about 2 times as viscous as water. Diseases like diabetes can also increase the viscosity of blood, therefore making it hard to penetrate some areas of the body. Osmolarity is the pull of the blood for water to come in. If it's too high, you absorb too much water, therefore the volume is high, the pressure is high, and blood pressure is also a big factor and will cause a disease. If it's too low, the water cannot be attracted back to the vessel, they will end up as edema. So it has to be the optimum osmolarity so that we have the right amount of water in the blood vessel. Protein, like we said, is a major factor to keep and attract water inside the blood vessel. If you have a condition of hypoproteinemia, where you have, as we break this down, emia means blood, hypo means below, and protein is the root word. So in the blood, there is low protein. That's what it means. It happens when the diet itself has no protein, as in severe starvation. If the liver is or kidney is deceased, for example, the liver is not functioning well, then it's not able to produce the plasma proteins. Or if the kidney is not functioning well, then it may be letting go of the proteins instead of keeping them. So that's why you're low in protein. Severe burns also would dictate a lot of protein deficiency because of the situation. There's also a nutritional condition or uh, physical condition called kwashiorkor. If children are fed mostly carbohydrates and not much protein, they will end up swelling in the intestines because the water stays in dependent areas like the intestines. So this is like edema of the abdominal cavity. We call ascites, right? There's not much protein in the diet, so you see their muscles are not well developed, or at least a good quality. And sometimes it's the situation. Protein is hard to get, and they will feed them with some other high caloric food like carbohydrates because it's what they have. So that's also a effect of malnutrition. Blood is produced in the bone marrow. As an adult, you can have 100 billion platelets, 200 billion red blood cells, and 10 billion WBCs every day, assuming you have a healthy setup, healthy bone marrow, and nutrition considered as well. The production of blood, especially the formed elements, is termed as hemopoiesis, and you have the yolk sac producing stem cells to begin the first blood, and it will continue on in the fetal bone marrow. And the liver stops producing blood cells at birth, but it usually catches up with the protein part of it. All through this time, the spleen will be involved in lymphocyte production. You have pluripotent cells, PPSC, colony-forming units, myeloid and lymphoid hemopoiesis. These are blood formation in the bone marrow for myeloid and blood formation in the lymphatic organs in the lymphoid. Okay, and this is in the beginning part, huh? Let's go to erythrocytes and know the function and structure of it, the hemoglobin, clinical measurements of that system, and then the life cycle of erythrocytes as well, huh? Here's a picture of the red blood cells, and as you can see, They are a little bit depressed in the center. In fact, when you see them in an actual slide, they would look like this, huh? Mm. And they are flexible and they can penetrate through small blood vessels. Here's another picture of them. These are the red blood cells, okay, erythrocytes, and these are the nuclei of the endothelial cells, which is the surface of the blood vessels. This is the capillary as a whole with the red blood cells. This is highlighting only the red blood cells, and this is highlighting the walls or the endothelium of the blood vessel. Red blood cells are known to carry oxygen. Tissues pick up also carbon dioxide, bring it to the lungs. Huh? Red blood cell is disc shaped. All organelles are not present and lost during development. They depend on anaerobic fermentation for the ATP that they need. That's also good because if they're anaerobic, they don't need the oxygen that they actually carry. Red blood cells also lack DNA and nucleus, so there's no protein synthesis or mitosis. As you can see, that's why it has less volume. Blood type is determined by surface glycoproteins and glycolipids uh, around the red blood cell. And we will deal with this and discuss this at the second part. Major transport of gas is important. The organelles in the red blood cells help to make this happen. Hemoglobin is very important, huh? Because of this, we're able to load up with oxygen. We also have carbonic anhydrase in the cytoplasm outside the red blood cell, which help us to convert carbon dioxide so we can transport it all the way to the lungs. Hemoglobin molecules consist of four protein chains or globins, which helps it to carry and be affinitive to the oxygen. You can have red blood cells, but if they don't have functional hemoglobin, you also will not be able to load up with oxygen and therefore will have the symptoms of anemia also. So these are the hemes, one at the center, and these are your fourth degree or quaternary structure of proteins. Huh? So it's a combination. That's why it's called hemoglobin. Huh? Heme and globin, the protein. Red blood cell count and hemoglobin concentration indicate the ability to load up with oxygen. That's why we measure hematocrit. Hematocrit is the formed elements with respect to the whole blood volume. So as we saw in the picture, the formed elements and the plasma. So with respect to the column of blood, how tall or how much is the formed elements with respect to that. In females, usually it's 36 to 46% of the whole column of blood that we sampled. In male, it's usually 38 to 48, a little bit more. Red blood cell count is also important because it tells us if there is sufficient transport of oxygen. Values are usually lower in women because androgen stimulates more red blood cell production in the male, and women would have their monthly periods. And plus, the more fat you have, the less hematocrit you do also perceive or have. Erythropoiesis is RBC production. A million RBCs are produced per second in a healthy adult. The red blood cells stay for about 120 days. After that, they really look worn out, and the spleen will be able to uh, take note and identify those who are have been for about that time, 120 days, and it will destroy it. All right. Anytime that we have low red blood cells, the system will alert the bone marrow and it will release more red blood cells. First committed cell is the erythrocyte CFU or colony forming units, which then will have a receptor for EPO. EPO comes from the kidney. EPO tends to stimulate the bone marrow, so it will produce and release red blood cells. Nucleus is discarded to form a reticulocyte. And the reticulocytes are basically the young red blood cells. So in visual, you see this as the hematopoietic stem cell, where all of this began. It becomes a CFU, and from there, the erythroblast becomes a reticulocyte, a young red blood cell, and then a mature erythrocyte. If a patient was taken, a blood sample was taken, and you see a reticulocyte, that means the young ones are being led to the battle too early. It's either they are not converting to mature red blood cells, or there's a bleed and you're using a lot of red blood cells, and now you have to call in the young ones, if you would. So we have to investigate. Iron metabolism starts when you have your diet, right, where it will be converted to another form of iron, gasoferritin. It will now be transported by transferrin in the blood plasma, and it will be deposited in the liver so we can use it for uh, making red blood cells, or at least completing the function of the red blood cell. And the remaining is distributed to other organs for use. Dietary iron is also important, definitely, and that uh, is a main source of our iron. The liver binds the apoferritin to create ferritin for storage of iron, which can be used in the future. Folic acid, right, is also dependent on this. Vitamin C and copper are important ingredients to make a healthy hemoglobin. A drop in red blood cell causes hypoxemia, which is detected by the kidney because the kidney is responsible for the blood passing through it. And when that happens, erythropoietin is released by the kidney, specifically your JG cells, juxtaglomerular cells, which will stimulate bone marrow to release red blood cells. If you have low levels of oxygen, hypoxemia will stimulate this condition to happen. High altitude, which is um, carrying thin quality of air, will make the body realize that there's less oxygen, and therefore will trigger the production of erythropoietin, abbreviated as EPO, huh? And therefore will make the body produce extra red blood cells. When you exercise, you also stimulate red blood cell production. Loss of lung tissue will trigger a situation like you are in hypoxemia, when that happens, again you will pick up and trigger the production of erythropoietin, and again the bone marrow will release red blood cells. So in visual presentation, any factor that puts you in hypoxemia will trigger the kidneys to produce erythropoietin. The kidney produces about 85% erythropoietin, and the liver produces about 15%. This will trigger the bone marrow to release red blood cells. Red blood cells are assumed to be available now to carry more oxygen. So the increase in red blood cell, we call that polycythemia, which is present in situations where low oxygen happens. And we just mentioned the factors that causes this to happen. Red blood cells are shiny like new cars when they are first introduced into the system, but as they go through the blood vessels, as they go and carry oxygen, as they are used through the couple of days, they also get some wear and tear. In fact, they can be roughy a little bit, rough on the sides as they go through the blood vessel. When they get to the spleen will identify them, and the macrophages will destroy them. All right, that's also good because then there's quality control if you would think about it. The heme will be separated from the globin. The globins can be hydrolyzed so we can use them when they turn into amino acids again. Remember, they're proteins. The iron is removed from the heme, and the heme pigment is converted to biliverdin. This is all happening in the spleen, huh? The finished product of the spleen is the bilirubin. The bilirubin coming from the spleen is called unconjugated bilirubin. The word unconjugated means it needs one more step. What would happen is this unconjugated bilirubin, which is yellowish in color, need to get to the liver. The albumin will piggyback or transport this to the liver so it can be converted. And the liver will now produce conjugated bilirubin. Conjugated bilirubin. The liver will use this conjugated bilirubin to make bile. To make bile. Part of this can be released in the kidney, giving the yellowish color of kidney itself. Bile, on the other hand, when produced, may be stored in the gallbladder, and when released into the kidney, actually creates urobilinogen, which adds to the brownness of the feces. Okay, so it's important that we know this function and know how this is processed. Now, the pathology here is one, when the unconjugated bilirubin is produced by the spleen and it's supposed to be brought to the liver. If the liver is diseased or you have hepatitis A or hepatitis B, E, C, or there's liver cirrhosis, obviously the liver cannot work 100%. What will happen is the unconjugated bilirubin will not be conjugated and they will be in the blood, and in the blood they will stay and give that yellowish discoloration, right? This will show as jaundice. The yellowish discoloration, this unconjugated bilirubin in the young, can reach the brain and it can stay in the brain, which can lead to mental retardation. So jaundice is a sign of unconjugated bilirubin. Now, I would also mention that unconjugated bilirubin showing as jaundice can be either physiologic or pathologic. A newborn who comes out jaundice may have physiologic or pathologic jaundice. It's physiologic if the liver, the blood vessels, right, are not yet that mature, and it will take a few days for it to mature. So what we do is we put the newborn under Billy light. We cover the eyes and the genitals, and then expose them. Billy light, what it does is if the blood vessels reach the surface, if the blood reaches the surface, Billy light is able to conjugate this bilirubin, and therefore convert it from being the yellowish unconjugated. This helps the premature organs, or at least the mature, not the organs to mature, especially the liver. In 10 days, it usually clears up. If it doesn't clear up at that time, we may be dealing with pathologic jaundice. And pathologic jaundice is when the problem actually happened before the child was born. It could be a case of uh, hemolytic disease of the newborn, where the newborn actually may be a second child, and the first child was a positive uh, blood type, which if the mother is negative, she was made to form antibodies to the positive, and therefore the mother is now giving blood to the second child with antibodies against the second child's blood if the second child happens to be a positive also. And so this blood problem is happening while in the womb. See, um, that can be a sign of jaundice when the child is born. It could also be some other factor like bone marrow problems or liver problems that's happening during the 9 months period. Okay, and this may need more than just Billy Light. This is more of a major problem. Maybe even transfusion is needed. Okay, all right. So that's the life and death of the red blood cells. Increased red blood cell is called polycythemia. Now, when it is because of the bone marrow producing a lot of red blood cell, maybe because of cancer conditions, then we call it primary polycythemia. When it is because of conditions like being in a high altitude and you have thin air and you're forced to produce more red blood cell, those are secondary polycythemia. Dehydration, when the plasma has been lost and you will have more of the formed elements, gives you a picture like it's a too much red blood cell, but actually it's just a dehydration condition. Too much red blood cell is not also favorable because it would increase the blood volume and sort of weight of the blood, the pressure and the thickness. This can double the work of the heart. This can lead to blood vessels having a high blood pressure also. This can lead to the possibility of clotting, especially when you have a lot of red blood cell and they stagnate, right? They can easily get destroyed and it will lead to clots. Uh, when these clots move through the circulatory system, they will be considered and they're considered as embolism or emboli. When it blocks the next vessel smaller in diameter, then you have a stroke, or if it's in the heart, you will have a heart failure. So that's polycythemia, too much red blood cell. Anemia is the opposite when you have less red blood cell, inadequate erythropoiesis or production of red blood cell. This could be because the kidney is failing and not able to produce erythropoietin, which should have triggered the bone marrow, right, to produce red blood cell. This may be because you don't have enough iron. This may be because you have lack of vitamin B12, which is have had when you have the ability to pick it up. Have intrinsic factor, but for some reason, if you don't have intrinsic factor, you won't be able to make use or pick up vitamin B12, and you'll end up in pernicious anemia. Hypoplastic anemia is anemia secondary to the slowing of red blood cell. Aplastic may be complete cessation of producing red blood cell. Bleeding is another reason why you may have anemia. You're losing red blood cell through the bleed. Destruction of the red blood cell by any chemical, any reason, right, that may do it. The red blood cells being destroyed will also lower down the count of your red blood cell. If you have anemia, that means you don't have much oxygen, and therefore the tissue will be hypoxic, and there can be necrosis. Patient will definitely be weak, lethargic. There'll be shortness of breath because you need oxygen, and you'll be gasping for breath. The brain will also be the heart and kidney can also be affected. They're very dependent upon oxygen. The osmolarity of blood is reduced, therefore you'll have more fluid in the tissue, causing edema. If the blood viscosity is also low because the quantity of red blood cell is low, then the heart has to work double time, and that could lead to heart conditions. Okay, so on both sides there is some danger. Sickle cell disease. This is a hereditary defect that shows in the red blood cells. The alleles modify the structure of hemoglobin, makes hemoglobin S. And when that happens, the red blood cells become rigid, sticky, and sometimes pointed at the end, making the shape of a sickle shape. Now, these red blood cells are not malleable or do not uh, change much. And unlike the healthy red blood cells, they can bend, they can be flexible, they can penetrate the small blood vessels or even a regular sized blood vessel. Sickle cell, excuse me, may clump or stick or back up in an opening in a blood vessel. It may end up blocking the actual blood vessel. And when that happens, you will have failure of that organ. So in sickle cell, it can lead to kidney or heart failure, stroke because of the blocked vessel, joint pain, paralysis also, right? So it can be devastating. The good side of sickle cell, though, is because the red blood cells are shaped as such, parasites that is found in malaria cannot survive in them. So in a malaria-prone or endemic area, the individuals who have sickle cell have less complications from malaria, maybe not even affected by malaria, because the parasites cannot live long. They can't penetrate the red blood cells. They're sickle cell shaped. Okay, and the sickle cell looks like this, huh? And they don't change their shape much, so they can be flexible and can't enter blood vessels completely. Shaped like a sickle. All right, we'll take a break from here before we go into blood typing, huh? Okay, we'll resume in a few minutes and then we'll continue our lecture on blood.